and R69 is a highly supported monophyletic group with a
structure that is alike. In utilizing the OMPA region of the Chlamydophila species,
has a biphasic developmental cycle, meaning that it alternates between extracellular.
However, poorly supported branching within the group can be seen as well.
Chlamydia are binded intracellular organisms. They progress internally through eukaryotic cells within membrane-bound vacuoles known as inclusions.
Oh, I can make it right. Alright, alright. Oh, I can make it right.
Hi guys! Welcome to our oral presentation season 2. We are group 2 and first let me introduce myself. I am Francisco III, pero please call me Iko. And together with me is my laboratory partner.
I'm Adriana Grinto, and together with us are Halot Hot, Teresa Elica Joy, Haranilla, Jeffrey Troy, and Jimenez Jasper John. And our team will be presenting our preliminary phylogenetic identification of virulent Chlamydophila picorum strains. So, Iko, do you know a thing or two about Chlamydophila or in general STD?
Chlamydia and STDs. Well, I can share a thing or two about chlamydias and STDs. So let's go. Well, let us not be confused na ang chlamydia or HIV, they are different terms. Magkaiba sila. Because chlamydia, first of all, ang major differences nila is that chlamydia can be cured while ang HIV naman, it cannot be cured. Pero, it can be controlled. And another thing is that chlamydia can be a complication, HIV rather can be a complication from chlamydia. And speaking of HIV, if you guys know Magic Johnson, siya ay isang professional basketball player, well-retired basketball player sa kapanahonan ni Michael Jordan. He was diagnosed with HIV at the age of 31. And now, 61 years old na siya. You might be thinking na baka patay na siya or mahina na siya. Well, actually, no. He's well and alive. And because, like I said, HIV can be controlled. So, ayun lang naman ang aking ma-share about sa chlamydia and STDs. So, let's go!
For the contents of the report, it is divided into 5 parts. This is to say for the abstract, introduction, materials and methods, discussion and results, and of course the conclusion. So let's proceed with the first one, or the first part, which is the abstract. Chlamydia are binded intracellular organisms. They progress internally through eukaryotic cells within membrane-bound vacuoles known as inclusions. And these inclusions have a distinctive biphasic developmental cycle. They take turns between extracellular and intracellular structures. And for the scope of the study, it is only limited within the OMPA, or the outer membrane protein gene region. It was used for the analysis and formulation of phylogenetic trees, of course through the usage of multiple tree-building algorithms. And the tree-building algorithm used were neighbor joining, maximum parsimony, and maximum likelihood. And for the results, neighbor joining and maximum likelihood showed a moderately supported bootstrap values. However, maximum parsimony in general is only fairly supported. It is due to the weakly supported scores for their few lineages. And moreover, for the multiple gene regions, OMPA, or the multiple outer membrane protein gene region, suggests that it is a fine molecular marker or indicator for succeeding epidemiological studies and investigations for Chlamydophila picorum. And that's all for the abstract. Take the floor, Troy. Thank you.
Thank you, Iko, for summarizing the abstract. Next slide, please. Chlamydia is a disease-causing bacterial phylum that are obligate, intracellular, non-motile, and gram-negative. What's unique about this Chlamydia is that it has a biphasic developmental cycle, meaning that it alternates between extracellular and intracellular forms. Analysis of 16S RNA divided the phylum into 2 genera, namely the Chlamydia and Chlamydophila. Seen in the PowerPoint presentation are the pictures of Chlamydia and Chlamydophila. Chlamydia species have gram-negative cell wall that is made up of an inner cytoplasmic membrane and an outer cytoplasmic membrane. The outer cytoplasmic membrane contains the major outer membrane protein, or MO, which is encoded by OMPA, or the outer membrane protein A. The OMPA is used for differentiating Chlamydia species, and one example would be the study of Fitch plus 2004, wherein OMPA was used to sequence 725 C. trachomatis cases in a Swedish county. Next slide. Chlamydophila picorum is another species of Chlamydia, and it is the topic of the study. As you can see, its picture is shown in the PPT slide. It has been documented to cause various diseases to cattle, ruminants, swine, and koalas. Non-pathogenic strains have also been documented. There are currently no typing methods that can differentiate the pathogenic and non-pathogenic strains of C. picorum. The goal of this study is to see whether OMPA is a good molecular marker for the differentiation of the two strains of C. picorum.
Thank you, Troy. Now for our materials and methods. The nucleotide sequence data of 18 Chlamydophila strains were retrieved from the provided accession numbers on the phylogenetic study of virulent Chlamydophila picorum strains as the main reference for this phylogenetic analysis. In this study, only the outer membrane protein A or OMPA gene region was utilized. The strains were identified to be as flashed in the screen. Due to inaccessible data for E58 and the outgroup Chlamydophila abortus S26 from the main reference, they were completely omitted from this study. The alignment of the sequences was performed using muscle, and the construction of phylogenetic trees neighbor joining maximum likelihood and maximum parsimony together with the analysis were conducted using the Mega5 software. The best fit model for NJ, ML, and MT analysis was then found to be the Tamura 3 parameter with gamma distribution. Now for the nodal support for the analysis, this was assessed with 500 bootstrap replications. Now let's move on to our results and discussion, starting with Adriana Ginto.
Thank you, Tere. I'm Adriana, and let's proceed to the discussion and results. In phylogenetic analysis of 18 strains, the researchers took note of the given groupings from the main reference of the paper. The groups are as follows. For G1, 6 pathogenic strains, 2 encephalomyelitis strains isolated in the US, the E58 and UK's SBE, 2 abortive strains from France, AB10, and Tunisia, AKT. A 1 orchitis strain, VB2 from France, and 1 arthritis strain, LW679 from the US. For G2, 3 ovine intestinal strains, 2 from Ireland, W73 and R69, and 1 from France, IB5. For G3, 3 cup-ring intestinal strains from France, IC3, IC2, and IC4. For G4, 2 ovine intestinal strains from France, IB1 and IB2. 1 bovine intestinal strain from the UK, BE53, and 1 ovine conjunctivitis strain from Scotland, 824. The following strains, IB4, M14, and IB3, were not grouped in the reference paper. Now let's proceed to the first tree building method, the neighbor joining analysis. According to Grimm, bootstrap values are accepted values of confidence. There are generally accepted thresholds for good and moderate support. Bootstrap values that are greater than 70 or 80 is considered good, and bootstrap values that are greater than 50 or 60, but less than 70 or 80 is considered moderate. In neighbor joining analysis, the constructed tree is observed to be moderately supported with the support range values of 57 to 100. Starting from G1, it consists of SBE, AKT, W679, and AB10, which is considered as a highly supported monophyletic group that has a support value of 100. This is also evident in its highly supported relationship with its sister taxa couplet G4, consisting of A24, BE53, IB1, and IB2. With a 99 support value, and the said group having a moderately supported value of 77. The analysis also shows that its in-group, consisting of IB1 and IB2, is highly supported with a support value of 100. It is also evident that its relationship with its sister taxa couplet, consisting of one bovine intestinal strain, the BE53, and one ovine conjunctivitis strain, the 824, from the UK and Scotland, respectively, that has a support value of 97, has a moderately supported value of 77, and shows monophyly. For G2, consisting of W73 and R69, is a highly supported monophyletic group with a support value of 97. Its relationship with IB5 is also highly supported with a support value of 97. Notably, the relationship between the ungrouped strain, IB4, to the monophyletic group consisting of sister taxa couplet G1 and G4 shows moderate support with a support value of 57. Moreover, the monophyletic G3 consisting of IC2, IC3, and IC4, on the other hand, shows a highly supported value of 100. Its monophyletic in-group IC2 and IC3, however, shows a moderate support value of 61. In conclusion, utilizing the neighbor joining analysis, all four groups within 18 Chlamydophila pecorum specimens showed monophyly, and the constructed tree is moderately supported with a support range value of 57 to 100. Now let's proceed to the maximum parsimony to be discussed by Teresa Halothot.
Okay. Thank you, Adi. Now, in the maximum parsimony analysis, some branches of the constructed tree are observed to have poorly supported nodal value. Starting from G1, it consists of SBE, AKT, VB2, LW679, and AB10, which resulted to be a monophyletic group. This relationship is also highly supported with a support value of 100. However, poorly supported branching within the group can be seen as well. The relationship between LW679 and its sister taxa couplet has inadequate support value of 1. This can also be observed in a relationship between AKT and VB2. G2, consisting of W73, R69, and IB5, is a highly supported monophyletic group with a support value of 93, and in-group relationships between W73 and R69 reflect a 100 support value. Results also show G3 as a highly supported monophyletic group with IC2, IC3, and IC4, though the relationship between IC2 and IC3 exhibits poor support with a value of 2. G4 appears to have a moderate support of 53 with a highly supported in-group relationships between A24 and BE53 with a support value of 88, and IB1 and IB2 with a support value of 100. Overall, using the maximum parsimony analysis, all four groups within the 18 Chlamydophila pecorum samples are found to be monophyletic. However, support value of the tree reflects instability of the relationship within the groups.
Thank you, Tere. And now, I am Jasper John F. Jimenez to discuss to you the maximum likelihood analysis. In the maximum likelihood analysis, strong support values are seen within the tree. The monophyletic group of G1, consisting of VB2, LW679, AKT, SBE, and AB10, are strongly supported with its 100 support value. Its sister taxa couplet G4 is also strongly supported with a support value of 96. However, this group has an inadequate support value of 42. G4 may have a low support value, but its in-group is strongly supported with a support value of 69 between 824 and BE53, and 99 support value for IB1 and IB2. G2, consisting of W73, R69, and IB5, is shown to be a highly supported group with a support value of 98, with W73 and R69 being highly supported as well, with 100 support value. G3 shows monophyly among IC2, IC3, and IC4, with a highly supported value of 100. And for the next slide, the results show consistency in analyzing the relationship of the groupings wherein G1, G2, G3, and G4 are all monophyletic groups. The trees may vary in topology and nodal support values. However, they all propose a general structure that is alike. In utilizing the OMPA region of the Chlamydophylla species, it was suggested that more similarities may arise between strains with different areas or host species than isolates within the same area or host. This backs up the monophyletic relationships of the strains from different locations or host species within the studied groups. Results from the study of Muhammad suggest that OMPA genes are sufficiently capable to discriminate the strains of Chlamydophylla pecorum, and recommends that OMPA genes are combined with INCA and ORF663 genes to produce a more informative classification.
Thank you, Jasper. We will now be moving for the conclusion. Next slide. There are three things that can be concluded from the study. First is that G1, G2, G3, and G4 groups form a monophyletic group. The different phylogenetic analysis shows that these groups are monophyletic and highly supported because of their high bootstrap values. Aside from that, the maximum parsimony also resolved the polytomy between G1 and G3 groups. However, the arrangement is poorly supported due to low bootstrap values. Second is that OMPA is capable of differentiating the different strains. However, it is not enough to properly resolve the polytomic issues of the phylogenetic tree. And lastly, is that the results of this study agree with the results of the study of Muhammad et al. 2008. The researchers are able to replicate the neighbor joining tree topology present in the study. And aside from that, Muhammad et al. Also recommends that OMPA should be combined with INCA and ORF663 to produce a more informative results. That's all for the conclusion. Let us now give the floor to Adriana and Francisco.
Thank you for that insightful conclusion, Troy. And here are the group's references for additional readings and citations used for the images. And this is where our group presentation about preliminary phylogenetic identification of virulent Chlamydophylla pecorum strains. Once again, I'm Adriana Ginto, and together with me is my lab partner,
Sidro, and along with us is Halot Teresa, Haranilia Troy, and Mr. Jimenez Jasper. So dito na nagtatapos ang ating oral presentation, guys. Thank you, and abangan natin ang season 3. So tara na, busy pa kami sa aming laboratory. Ikaw ba, Adi, busy ka ba dyan?
sobrang busy. Like, hindi ko na alam kung susunod kong gagawin. Char.
Ayun, guys. Thank you. Bye-bye. Season 3 Season 3.
I can make it right. Alright.